Okay. So we're about to solve May, June 2024, paper 1, variant 2. A prokaryotic cell, which is 1 micrometer in diameter, is magnified 50,000 times in an electron micrograph. What is the diameter of the cell in the electron micrograph? So this is your actual size. And this is your magnification, m equals to I upon A. Now, all the answers are in millimeters, so we will convert the micrometer value into millimeters. So 1 micrometer would be 0.1 into 10 to the power minus 3 millimeters. 450,000 into 1 into 10 power minus 3 would give us the image size, which would come out to be 50 millimeters, which would be option number C. The diagram shows a plant cell with some label structure, which label structures are bound by a double membrane. So P is nucleus. Yes, nucleus has a double membrane. Q is chloroplast. Chloroplasts as well have a double membrane. R is your cell membrane, which does not have a double membrane actually. And S is your vacuole, which is again made up of single membrane. So your correct answer would be A, P, and Q. Which size of ribosome is found in mitochondria and typical prokaryotic cells? 70S ribosomes. Which row about typical prokaryotic cells and typical animal cells is correct? The key is correct for typical prokaryotic cells and typical animal cells. Cross suggest not correct for both cells, but correct for either typical prokaryotic cells or typical animal cells. If there is a tick, it means it is present in both. If it is a cross, it means it is not present in both. Lysosomes present for the breakdown of old organelles. Lysosome is not present in prokaryotic cells, so this has to be a cross. ATP is produced by the cell. Now, ATP is produced by prokaryotes as well, although they do not have a mitochondria, but still they are capable of aerobically respiring. And even if they can anaerobically respire, in both the cases, they will break down whatever organic molecules they consume into ATP to release ATP. So, option number C is the correct answer. Which row is correct for the structures present in typical plant cells and typical animal cells? Plasmodesmata is not present in animal cells. Golgi body is present in animal cells. So A is out, B is out. C, centriole. Centriole is not present in plant cells. That is correct. Centriole is present in animal cells. So this option seems to be the answer. Tonoplast, not present, not present. Tonoplast is present in in plant cells, and hence D is also incorrect. Which row is correct for cellulose? Rotation of alternate monomers by 180 degrees? Yes. So it has to be a tick. Shape of molecule has to be unbranched. It's a linear molecule. Hydrogen bonds between molecules? Yes. This is option number D. Okay. Which statements about peptide bond formation are correct? The bond formation occurs between a carbon of one amino acid. So yes, carbon of one amino acid and a nitrogen of the next amino acid after the amino acids detach from tRNA. Now this part of the statement is incorrect. हमारे पास पहले peptide bond form होता है between consecutive amino acids and then the amino acid is detached from tRNA. तो पहले bond formation होती है, उसके बाद detachment होती है. So the order is reversed. The bond formation occurs at the ribosome while the amino acids are still attached to tRNA, and it is a hydrolysis reaction. Now, this part of the statement is incorrect. That is a condensation reaction. The bond formation is important for growth of an organism, and when the bond forms, the water molecule is removed. Yes, option number D is the correct answer. The diagram shows naturally occurring D-glucose and a form of glucose that can be synthesized in the laboratory known as L-glucose. The enzyme glucose oxidase catalyzes the oxidation of D-glucose. The enzyme cannot catalyze the oxidation of L-glucose. Which statement about L-glucose explains this? L-glucose does not fit the active site of glucose oxidase, seems to be a correct explanation. L-glucose has a different structural formula to D-glucose. L-glucose is a synthetic sugar. L-glucose is the mirror image of D-glucose. These explanations are not correctly explaining why L-glucose is no longer, L-glucose cannot be catalyzed by or oxidized by glucose oxidase. The reason that we suggest for an enzyme not catalyzing a reaction on a particular substrate has to be the fact that the enzyme and the substrate do not have a complementary shape. So A would be the correct answer. Tests for biological molecules were carried out on three solutions. Each solution contained only one type of the biological molecule. The observations were as follows. So the first solution we had Benedict's test, blue to orange. This means a reducing sugar is present. Benedict's test after acid hydrolysis, blue to red. So after hydrolysis हमारे पास reducing sugar present है. Biuret test, blue to purple. That means that protein is present. So yeah. So after the hydrolysis, we have reducing sugar. Before hydrolysis, we did not have a reducing sugar. Which solutions would contain either sucrose or amylase? So amylase is what? Amylase is a protein. It is an enzyme. So this would be what? This would be solution 3. And for sucrose, we would say that sucrose, because we're doing an acid hydrolysis, it is a possibility that before this acid hydrolysis, we had sucrose present. So there is a possibility of sucrose being present in solution number 2. So option number C is the correct answer over here. Which row describes the expected effect on Vmax and Km when a competitive reversible inhibitor is added to an enzyme catalyzed reaction? The Vmax is always reached in the presence of a competitive reversible inhibitor. हमारे पास Vmax की जो value है, that does not change when we are adding a competitive inhibitor. ठीक है? So there is no change in the value of the Vmax. However, there is a change in the value of Km, the substrate concentration at which our enzyme's activity is 50% of its maximum rate. That does change. And how does that change? It changes in a way कि हमारे पास जो Km value है, it increases. And hence option A is the correct answer. Yeah, non-competitive inhibitor does this decrease the Vmax value, but over here the question is about the competitive inhibitor now. Right. The graph shows the effect of substrate concentration on the rate of reaction of three enzymes X, Y, and Z. So we have the rate of the reaction on the y-axis and we have substrate concentration on x-axis. What is the correct order of affinity? Correct order of affinity of these enzymes for their substrates, starting with the enzyme with the highest affinity. Okay, we predict affinity on the basis of Km value. What is the Km value? It is the substrate concentration at Vmax upon 2. So हमारे पास सबका Vmax है. इस Vmax से हम Vmax upon 2 के जो Km values हैं उनको figure out करते हैं. So enzyme X जो है उसका Vmax is 1500. Half of 1500 is 750. So this would be the Km for enzyme X. Now, if we look at enzyme Y is somewhere around, let's say 2200 अगर हम इसका Vmax ले लें. So the half of 2200 would be 1100. So 1100 would be somewhere here. Then if we talk about enzyme Z, if we take this as maybe 200, अगर हम इसको 200 ले लें. So let's say हमारे पास 100 जो है, यानी इसका जो half है, it's somewhere here. And this is basically its Km. So Km of Z, and then we have Km of Y. So this is basically X, Z, Y, which is option number B. Yes. Which row correctly identifies the weak and strong bonds in the tertiary and quaternary structure of a typical protein? When we're talking about the four bonds, disulfide, hydrogen, hydrophobic, ionic, it is very important to understand that the strongest bond is only the disulfide bond when we are talking with respect to enzymes. All the other three bonds are considered to be weak bonds, and hence option B is the correct answer. Which row correctly describes hemoglobin? Four polypeptide chains, each containing a heme group. Yes. Iron ions can associate with oxygen, forming oxyhemoglobin. Yes. In each chain, hydrophobic R groups of amino acids point towards the center of the molecule. Yes, because this is the reason why hemoglobin is a globular protein. At 50% saturation, two oxygen molecules are transported by the hemoglobin molecule. All these four statements are absolutely correct. Option A is the correct answer. Which process always takes place without the involvement of energy from ATP? Now, active transport is an active process. It requires ATP. Endocytosis and exocytosis are both active processes. All of these need ATP. And hence, option D is the correct answer because it is a passive process. The diagram shows the entry of a molecule X into a cell. Which row shows the property of molecule X and the effect of the concentration of ATP in the cytoplasm on the rate of entry of molecule X? Now, looking at this diagram, can you guys tell me whether this is a channel protein or whether it is a carrier protein? Walaikum assalam. It's a channel protein, right? Now, channel proteins are responsible for facilitated diffusion, not active transport. So, हमारे पास जो concentration of ATP है, that would remain unaffected. So, my पास either B or D. And we know we need channel proteins for polar molecules, and that is why option number D is the correct answer. The electron micrograph shows some human blood cells. Which row correctly shows the net movement of water by osmosis and the water potential of the cytoplasm of cell X compared with the solution surrounding the cells? Now, if you look at cell X is basically a shrunken RBC. My पास normal RBCs are rather thicker, but over here we can clearly see कि हमारे पास ये जो RBC है वो shrink हो गया, इसकी हमारे पास crenation हो गई है. Now, crenation कब होती है? Crenation तब होती है जब हमारे पास सेल अपना water content lose करता है. If the cell is losing the water content, then the cell cytoplasm had a higher water potential in comparison to the surrounding solution, and therefore the water moved out of the cell. And thus, option C is the correct answer. Is this clear? हमारे पास ये कहते हैं they are talking with respect to the fact कि हमारे पास osmosis क्यों हुआ. Osmosis, yeah, exactly. They're talking before osmosis that what was the reason or what was the water potential gradient which caused osmosis. ठीक है? Okay. A red indicator solution was mixed with agar and the resulting solid was cut into small cylindrical blocks. The blocks were placed in an acid which turned the indicator yellow, and all other variables were kept constant. The dimensions of the blocks are shown. So they've given us the height of each block, they've cylindrical block, and they've given us the diameter. They've also given us a formula of surface area of a cylinder, which is 2 pi RH plus 2 pi R squared, and the volume of a cylinder, which is pi R square H. And they're asking us which row shows the correct surface area to volume ratio for each block and the time taken for the block to turn yellow. Now, हमारे पास you have to understand यहाँ पर जो छोटा सा concept है, and I'm pretty sure because you guys have sat for your practical exam, you will be very much aware with this concept कि हमारे पास ये वाला जो test होता है इसमें हम देख रहे होते हैं how fast the process of diffusion is. Now, हमारे पास there is a red indicator solution, and what we're doing is that we're placing those blocks in an acid, and that acid turns the indicator yellow. हमारे पास क्या कहते हैं जो कलर चेंज है वो सबसे ज्यादा जल्दी कब आएगा? कलर चेंज सबसे ज्यादा जल्दी तब आएगा जहां पे सबसे जल्दी एसिड डिफ्यूज होगा. सबसे जल्दी एसिड कहां पर डिफ्यूज होगा? जहां पर सरफेस एरिया टू वॉल्यूम रेशियो सबसे ज्यादा होगा. So greater the surface area to volume ratio, faster time taken to change color, or in other words, we can also say um, less time taken for color change. jaldi ho jayega basically. Now for that, we'll have to calculate the surface area and the volume ratios. हमारे पास इसको पहले रेडियस में कन्वर्ट कर देते हैं. 3 millimeters, 6 millimeters, and radius is 8 millimeters. Now if you put these values in the calculator, तो अगर हम ब्लॉक 1 की बात करें, ब्लॉक 1 का जो वॉल्यूम है, that would come out to be 27 pi. I'm not converting the pi into, what do you say, the numeric decimal value, because eventually वो cancel out हो जाएंगे. Surface area जो है, that would come out to be 36 pi. Now when you would do the surface area to volume ratio, which is when you'll divide 36 pi by 27 pi, the surface area to volume ratio would come out to be 1.33. So C या D में से कोई आंसर होगा हमारा. Okay. Now over here, अगर आप बाकी की calculations ना करो, although I would suggest that do the calculations for being sure and confident in your choice of answer, but let's say जल्दी paper करना है या time कम है, whatever. If you, in this case scenario, you can actually do a smart move, which is that you have figured out your first block का surface area to volume ratio क्या होगा. And you can clearly see कि C और D में बाकी जो options हैं for block 2 and block 3, they are same. So you know your answer has to be C or D because आपने पहली calculation कर लिया. बाकी दो कॉलम्स बिल्कुल सेम एज इट इज हैं, कोई फर्क नहीं है सी और डी के सरफेस एरिया टू वॉल्यूम रेशियोज वाले कॉलम में. सो यू कैन इजीली फिगर आउट कि ब्लॉक 1, ब्लॉक 2, ब्लॉक 3 का सरफेस एरिया टू वॉल्यूम रेशियो सबसे ज्यादा किसका है, सबसे कम किसका है. जिसका सरफेस एरिया टू वॉल्यूम रेशियो सबसे ज्यादा होगा उसका सबसे कम टाइम लगेगा कलर चेंज में. So सबसे कम टाइम किसको लगेगा, ऑब्वियसली इसको लगेगा because it has the highest surface area to volume ratio. After that, this, and then the one with the least surface area to volume ratio would take the longest time, the most time for the color change to appear. And hence option C is the correct answer. अगर आप ब्लॉक 2 और ब्लॉक 3 की कैलकुलेशंस भी करोगे सर्फेस एरिया टू वॉल्यूम रेशियो की, यू विल गेट 0.67 एंड 0.5 एज द आंसर. हाँ, अगर हमारे पास ये कहते हैं डी और सी के जो ये ऑप्शंस होते हैं सर्फेस एरिया टू वॉल्यूम रेशियो के, तो आपको ब्लॉक 2 और ब्लॉक 3 का भी कैलकुलेट करना पड़ता. But over here, because they've made our life easier in this case scenario, there's no need to find out the surface area to volume ratio for block 2 and for block 3. Which metabolic processes will be very active in a cell that has just completed cytokinesis? ATP formation, yes, because ATP formation is simply what? It's your respiration. Now, DNA replication is something that only occurs in the S phase. And the cell that we're talking about has just completed cytokinesis. It means कि ये किस में होगा, अपने interphase में है cell. तो interphase के S phase में हमारे पास DNA replication होती है, which is not at the start. It is in the middle of G1 and G2. तो DNA replication नहीं होगी. Protein synthesis बिल्कुल होगा. Option B is the correct answer. Is this clear? Any questions about this? Okay. The diagram shows a typical mitotic cell cycle and the points in the cell cycle that has been reached by each of 4 cells V, W, X, and Y. Which row correctly identifies the cells that match the two descriptions? Acha, how ATP forms? ATP formation is basically a result of respiration, right? Respiration basically produces ATP. Now respiration हमारे पास सेल में थ्रू आउट हो रही होती है ना, इंटरफेस में तो हो रही होगी. So हमारे पास जो respiration and that would be resulting in ATP formation, which would be something we will be seeing in a cell that has just completed cytokinesis. Okay. Actually, which row correctly identifies the cells that match the two descriptions? DNA replication is complete, but the cell has not yet reached its maximum size. Now, DNA replication has been completed. That's point W. ठीक है। और हमारे पास सेल अपना मैक्सिमम साइज नहीं रीच किया, इसका क्या मतलब है कि हमारे पास सेल अभी और ग्रो करेगा। एंड G2 फेज इज व्हाट, वहां पे हमारे पास ग्रोथ हो रही होगी, प्रोटीन सिंथेसिस हो रहा होगा, लिपिड सिंथेसिस हो रहा होगा। सो W ऐसा पॉइंट है जहां पे डीएनए रेप्लिकेशन कंप्लीट हो गई है, एस फेज कंप्लीट हो गया। बट अभी भी सेल थोड़ा बहुत ग्रो करेगा, अपना साइज इंक्रीज करेगा। सो W इज द पॉइंट वेयर डीएनए रेप्लिकेशन हैज बीन completed बट द सेल हैज नॉट yet reached its maximum size. Now, if we talk about the second column, preparation for microtubule formation is nearly completed, but chromosomes have not yet condensed. Chromosome condensation starts in prophase. Prophase is the first stage of mitosis, right? तो हमारे पास दैट हैज टू बी एक्स, माइटोसिस से जस्ट पहले, पीमैट स्टार्ट होने से जस्ट पहले हमारे पास जो सेंट्रियोल्स होते हैं वो रेप्लिकेट कर जाते हैं, ऑपोजिट पोल्स पे चले जाते हैं। सो हमारे पास माइक्रोट्यूब्यूल फॉर्मेशन की प्रिपरेशन हो जाती है बिफोर माइटोसिस स्टार्ट्स। माइटोसिस जब स्टार्ट होगा, प्रोफेज जब स्टार्ट होगा, तब कॉन्डेंसेशन स्टार्ट होगी। सो ऑप्शन डी इज द करेक्ट आंसर। The graph shows the mean length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fibers? This is a very interesting question. What do you guys think? I would like if you guys read this, go over the graph, and then tell me the answer. Okay. What about Mutahar and Fiza? Sorry, Farhat. Yeah, it'll be D. Why would it be D? बिकॉज मीन लेंथ का क्या मतलब है कि हमारे पास जो स्पिंडल फाइबर्स हैं जो एक पोल से दूसरे पोल एक्सटेंड कर रहे हैं, उनकी जो एवरेज लेंथ है वो कितनी होगी। नाउ द स्टेटमेंट इज सेइंग व्हिच रीजन ऑफ द ग्राफ शोज व्हेन ऑल द सेंट्रोमेयर्स हैव detached from the spindle fibers. Now this is the point where anaphase has completed. एनाफेज में क्या हो रहा होता है, हमारे पास जो स्पिंडल फाइबर्स हैं वो कॉन्ट्रैक्ट कर रहे होते हैं, और कॉन्ट्रैक्ट करते करते वो बिल्कुल इतने क्या कहते हैं थोड़े से रह जाते हैं। बाय द टाइम द सेंट्रोमियर्स वुड डिटैच, हमारे पास जो माइक्रोट्यूब्यूल फाइबर्स हैं दे वुड नो लॉन्गर एग्जिस्ट, या वो बहुत थोड़े से अमाउंट में एग्जिस्ट करेंगे। एंड हेंस आवर ऑप्शन, द करेक्ट ऑप्शन वुड बी the mRNA codons ACU, ACC, ACA, and ACG all code for the same amino acid threonine. Which anticodons could specify an amino acid other than threonine? Okay. सो हमें आइडेंटिफाई करना है इस पर्टिकुलर अमीनो एसिड के अलावा वाले कोडॉन्स। हमें पता है ये चार जो कोडॉन्स हैं दे आर फॉर थ्रियोनाइन, ठीक है। आई डोंट नो इफ इट्स थ्रियोनिन और थ्रियोनाइन, बट व्हाटएवर दिस अमीनो एसिड इज, इसको कोड करने के लिए हमारे पास एमआरएनए कोडॉन्स चार हैं। तो ऑब्वियसली इसके एंटी कोडॉन्स भी चार होंगे। सो द एंटी कोडॉन्स वुड बी यू जी ए, देन वी वुड हैव यू जी जी, U GU, and UGC. So these are the four anticodons for this particular amino acid. We have to identify anticodons for an amino acid other than this one. So हमारे पास इसके वाले anticodons को हम cancel out कर देंगे. So this is out. And हमारे पास क्या कहते हैं और कौन सा out होगा? UGC, sorry, not UGG. UGC will be out. So 1 and 2 would specify an amino acid other than this particular amino acid, and hence option B is the correct answer over here. Which bond formation does DNA polymerase catalyzes? We know that DNA polymerase is responsible to form the phosphodiester bonds to complete the sugar phosphate backbone. So not hydrogen bonds for sure. A and B are out. Between C and D, the correct answer would be D, because हमारे पास जो phosphodiester bonds बनते हैं that they are between nucleotides, not between bases. Bases के दरमियान हमारे पास hydrogen bonds बनते हैं. In eukaryotes, the RNA molecules formed during transcription are modified by the removal of the non-coding sequences, which we know are the introns. This is followed by the join together of coding sequences to form mRNA. What are the coding sequences also called? The coding sequences are called exons. Which row correctly identifies sinks for sucrose transported by mass flow in plants? Now, again, a very interesting question. I would like if you guys predict an answer for this. Yes. A is the correct answer. Now, wow. Now the question is, why A is the correct answer? अच्छा, हमारे पास root storage organ would definitely be a sink. Sink is basically that place which receives the sugars, right? तो हमारे पास जो storage organ है उसका काम क्या होता है कि वो sugars जो हो गए, food molecules जो हो गए, उनको store रखे for any drought conditions or for later use जब हमारे पास enough food supply ना हो तो ये जो storage हमारे पास molecules हैं इनको break down करके energy produce की जा सके. So root storage organ would definitely be a sink. Now, when they say growing leaf bud or growing shoot tip, now anything that is growing, which is not yet matured, it is not photosynthesizing. It still needs nutrition for itself because it is in its growth phase. हमारे पास जो mature leaf होता है, a mature leaf is capable for photosynthesis. जब वो एक लीफ बड होती है, जब वो क्या कहते हैं properly mature leaf जब तक वो नहीं बन जाता, तब तक that leaf bud, that immature leaf would be a sink. It would require sucrose from a particular source. So during the growth phase, a leaf is not able to photosynthesize itself, and therefore it also acts as a sink. And growing shoot up again would be a sink as well, and hence option A is the correct answer. The diagram shows a transverse section through a transport tissue in a plant. Which row correctly identifies cell 1 and cell 2? Now again, I would like you guys to tell me what do you think is cell 1 and what do you think is cell 2? Right. So cell 1 is a companion cell and we can clearly see the sieve plate, the sieve pores in cell number 2 and hence option A is the correct answer. Which statement correctly describes the movement of solutes in the symplast pathway? Cell surface membranes regulate the selective absorption of solutes into the symplast pathway. This seems to be a correct answer. Plasmodesmata control the movement of solutes from the symplast pathway to the apoplast pathway? No. Plasmodesmata is only responsible for the movement from the symplast pathway to the symplast pathway only. The symplast pathway transports dissolved mineral ions from the soil that cannot be transported by the apoplast pathway. Incorrect. It is the other way around. हमारे पास जो क्या कहते हैं चीजें जो सेल सरफेस मेम्ब्रेन को क्रॉस नहीं कर सकती वो सेल वॉल को क्रॉस कर सकती हैं. So yeah, हमारे पास this is basically a reverse situation that they've given us. The movement of solutes through plasmodesmata in the symplast pathway is prevented in the endodermis by suberin. Again, this is the opposite that they've mentioned. Suberin blocks the apoplast pathway, not the symplast pathway. Which statement helps to explain why water molecules are forced to move through xylem vessel elements as a consequence of transpiration? Water molecules form hydrogen bonds with cellulose in the walls of xylem vessel elements in a process known as adhesion. Water molecules form hydrogen bonds with neighboring water molecules in a process known as cohesion. Water molecules form ionic bonds with dissolved mineral ions, which helps to keep the water molecules together in a continuous column. Water has a high latent heat of vaporization and this prevents the evaporation of water in the xylem vessel elements. C and D तो हम ऐसे eliminate करते हैं. A and B में हमारे पास B would be the correct answer. Why? Because हमारे पास जो model है, that model is known as the cohesion tension model of movement through the xylem vessel. तो हमारे पास जो cohesion है और जो tension, this tension is basically the transpirational pull. These are the two causes or the two forces which are responsible for the movement of water through the xylem. The diagram shows the internal structure of the mammalian heart, which letter identifies the location of the atrioventricular node. That is B. Not A, it is B. If you look at the pictures, अगर आप Google करो अभी, तो आप clearly देख सकते हो कि हमारे पास जो avian होता है, that is basically present हमारे पास बिल्कुल towards the center of the heart. So it's at a boundary of the atria and the ventricles, more towards the atria side. और वो हमेशा आपको सेंटर में नजर आएगा, सेंटर ऑफ द हार्ट की तरफ नजर आ रहा होगा, मीडियन सेप्टम के पास नजर आ रहा होगा. So B is the correct identification of the location of the AVN. The diagram shows pressure changes in the left side of the heart during the cardiac cycle. What happens in the heart at X? Now, point X is where the ventricular systole starts. ये स्टार्टिंग पॉइंट ऑफ वेंट्रिकुलर कॉन्ट्रैक्शन है। इस पॉइंट पर हमारे पास जो AV valve है that closes. Why does the AV valve close? It closes so that there is no backflow of blood into the atria as the ventricular starts to contract, as the ventricle starts to contract. So A is the correct answer. ठीक है? Okay. Which components of blood are present in tissue fluid? So phagocytes are present in tissue fluid. Some proteins, हमारे पास small proteins are actually found in the tissue fluid. Sodium ions again are found in tissue fluid, and hence option A is the correct answer. Okay. Now for this question, I would suggest कि जब भी ऐसा कोई question आए, आप diagram draw करो, and that diagram would help you understand the question and answer the question correctly. The question is saying in the lungs, movement of dissolved carbon dioxide out of the capillaries occurs in one of the two ways, either by diffusion through the endothelial cells of the capillaries or by leakage through pores in the endothelial cells of the capillaries. What is the minimum number of times that a carbon dioxide molecule that has been transported to the lungs in a red blood cell must cross a cell surface membrane to reach an airspace in an alveolus? Now, ये आपके पास एयर स्पेस है, ये आपकी कैपिलरी है इन द रेड लाइन दैट आई हैव ड्रॉन, ये मैंने कैपिलरी दिखाई दी, और कैपिलरी के जो एरियाज हैं जहां पर हमारे पास ब्लू कलर में ड्रॉन एंडोथेलियल सेल्स आपको नजर नहीं आ रहे, वो उसके पोर्स हैं. डार्क ब्लू में आई हैव ड्रॉन द एंडोथेलियल सेल्स ऑफ द कैपिलरीज. Now they're asking us the minimum numbers of cell surface membranes that the carbon dioxide molecule has to cross while it moves from the RBC into the airspace. Now, हमारे पास कार्बन डाइऑक्साइड मॉलिक्यूल का है, कार्बन डाइऑक्साइड मॉलिक्यूल है RBC के अंदर, ठीक है. Now, if it is moving outside of the RBC, तो उसके दो पाथवेज हैं. एक पाथवे ये है कि वो RBC को क्रॉस करे, ये इसकी पहली, इसने RBC की मेम्ब्रेन को जब क्रॉस किया, इसकी ये पहली मेम्ब्रेन थी. नाउ हमारे पास एंडोथेलियल सेल्स नहीं है, तो एंडोथेलियल सेल्स को इसको क्रॉस करने की जरूरत नहीं है. ये अब एंटर करेगा हमारे एपिथेलियल सेल्स ऑफ द एल्वुलाई में. ये इसकी सेकंड एंट्री हुई, सेकंड मूवमेंट हुई अक्रॉस सेल सरफेस मेम्ब्रेन, और ये उसकी थर्ड सेल सरफेस मेम्ब्रेन इसने क्रॉस की टू एंटर द एयर स्पेस. So the minimum number of cell surface membranes that a carbon dioxide molecule would cross is 3. मैक्सिमम क्या होती? मैक्सिमम हमारे पास होती, एक ये मूव करता, क्या कहते हैं, RBC की, फिर हमारे पास क्या कहते हैं, एंडोथेलियल सेल्स की पहली दफा, फिर एंडोथेलियल सेल्स की दूसरी दफा, एपिथेलियल सेल्स, and this would be 5. So the maximum number would have been 5, minimum would have been 3. Max 5, minimum 3. Is this clear? Any questions? How 5? अच्छा, एक second. Let me explain. See, ये एक RBC है, ठीक है. This is an endothelial cell, और उस endothelial cell के ऊपर we have epithelial cell. तो ये epithelial cell है alveoli का, ये हमारे पास endothelial cell है, and this is RBC. So RBC की membrane, endothelial की membrane दो दफा, and then twice epithelial. So yeah, that's how 5. What maintains the steep concentration gradients needed for successful gas exchange in the lungs? Airflow in the alveoli is in the opposite direction to blood flow in the capillaries. This happens in fishes. हमने ये fishes का जो counter current movement था, हमने उसमें पढ़ा था. Humans में ऐसा नहीं होता. So lungs में ये नहीं होता, ये gills में होता है. Blood arrives in the lungs with a lower oxygen concentration and a higher carbon dioxide concentration than the air in the alveoli. That is correct. Blood is constantly flowing through and out of the lungs, bringing a fresh supply of red blood cells. Yes, option number D is the correct answer. Any confusions? Any questions? Where is cartilage tissue always found in the human gas exchange system? Bronchi and trachea. Scientists compared the density of goblet cells in the lungs and the density of mucus in the lungs of three groups of people: people who do not smoke and do not have lung disease, people who smoke tobacco but do not have lung disease, people who smoke tobacco and have lung disease. The results are shown in the table. So the non-smokers have the least number of goblet cell density and they also have the least mucus density. And smokers who do not have lung diseases have the maximum number of goblet cell density and they have the maximum amount of mucus density as well. Smokers with lung disease are in the middle. They have goblet cell density greater than non-smokers, but less than the smokers who do not have lung diseases. And again, उनका mucus density भी middle में ही है दोनों cases क्या. What is indicated by these data? There is a positive correlation between density of goblet cells and density of mucus. Positive correlation का मतलब ये है, if one quantity increases, for example, goblet cell density increases, then the mucus density also increases. This is a positive correlation. Negative correlation का मतलब ये होता है एक बढ़ता दूसरा कम होता. So we can clearly see that whenever the goblet cell density increases, हमारे पास जो mucus की density है वो भी increase कर रही है. So yes, there is a positive correlation between these two quantities. Lung disease results in an increase in goblet cell density. Now this is incorrect. Why? Because those who do not have lung diseases have more goblet cells in comparison to the ones which have lung diseases. So हमारे पास lung disease does not result in an increase in goblet cell density. Instead, it is the opposite. So actually we have our answer, option number C. 3 भी पढ़ ही लेते हैं. There is an association between tobacco smoking and an increase in mucus density. Yes, हमारे पास ये smokers हैं. अगर हम इनके mucus density को compare करें with non-smokers, so the smokers have a greater mucus density. Which disease does Mycobacterium bovis cause? Tuberculosis. An antibiotic inhibits the formation of crosslinks between the molecules that form cell walls in bacteria, which statements explain why bacteria are killed by the antibiotic. A bacterial cell is destroyed by osmotic lysis. Yes, if there is no cell wall to support the bacterial cell, then yes, उसका osmotic lysis हो सकता है because of a weak or no cell wall. सेल्यूलोज मॉलिक्यूल्स कैन नॉट फॉर्म हाइड्रोजन बॉन्ड्स. इनकरेक्ट, क्योंकि हमारे पास बैक्टीरिया में सेल्यूलोज की सेल वॉल नहीं होती, पेप्टाइडोग्लाइकन की सेल वॉल होती है. अगेन हमारे पास क्या कहते हैं, द सेल वॉल इज नो लॉन्गर पार्शियली परमिएबल. सेल वॉल इज नेवर पार्शियली परमिएबल, हमेशा वो फुली परमिएबल होती है. सो 3 इज ऑल्सो इनकरेक्ट एंड ऑप्शन नंबर C is the correct answer. Is this clear? Any questions? Okay. This is a very annoying and a very stupid question, in all honesty. Scientists investigated the effect of increasing concentrations of an antibiotic on the development of antibiotic resistance in bacteria. So they're basically seeing अगर हम antibiotic concentration बढ़ाएंगे तो इसका effect क्या होगा on the percentage of resistant bacteria. So scientists grew 4 groups of bacteria and added a different concentration of antibiotic to each group. The number of resistant bacteria and the total population of bacteria were measured at intervals for 24 hours for each group. The graph shows the result. So ये हमारे पास control group है जिसको हमने कोई antibiotic नहीं दी है. And then we have 3 experimental groups. एक को हमने 90 mg per decimeter cube antibiotic दी है, एक को 215 mg per dm cube दी है, एंड एक को हमने 600 mg per dm cube ली है. ठीक है, हमारे पास जो straight black line है that tells us about the population of bacteria, the total population, and the dotted line basically tells us the resistant population. Which statements are correct conclusions that can be made from the results of this investigation? Increasing the concentration of antibiotic decreases the population of non-resistant bacteria at the end of 24 hours. अच्छा, अगर आप देखो क्या कहते हैं केस नंबर ये वाले में 215 mg per dm cube में, तो इसमें ऐसा हो रहा है कि हमारे पास 24 आवर्स में डिक्लाइन हमें नजर आ रहा है इन द रेजिस्टेंट बैक्टीरिया पॉपुलेशन. ऐसा हमें किस में नजर आ रहा है, केस नंबर 215 mg per dm cube वाले में. हमारे पास 600 पे रिलेटिवली कांस्टेंट लाइन है, रेजिस्टेंट बैक्टीरिया की, ठीक है। और ये भी थोड़ा सा क्या कहते हैं स्टार्टिंग पॉइंट से डिक्रीज हुई है। इफ यू कैन सी हमारे पास जो स्टार्टिंग पॉइंट है उससे अगर हम कंपेयर करें तो हमारे पास जो कॉन्सेंट्रेशन कह जो पॉपुलेशन है रेजिस्टेंट बैक्टीरिया की वो थोड़ी बहुत कम हुई है एट द एंड ऑफ 24 आवर्स। सो इस केस में भी हमें नजर आ रहा है कि थोड़ी बहुत जो हमारे पास रेजिस्टेंट बैक्टीरिया की पॉपुलेशन है इट हैज डिक्रीज्ड एट द एंड ऑफ 24 आवर्स। इसमें हमें ऐसा दिखाई नहीं दे रहा। तो बेसिकली हमने जब 90 से 215 पे इंक्रीज किया और हमने 600 पे इंक्रीज किया, सो वी कैन सी कि इंक्रीजिंग द कॉन्सेंट्रेशन इज समहाउ डिक्रीजिंग द पॉपुलेशन ऑफ द resistant bacteria. Is this clear? Can we say that statement number one is correct? एक सेकेंड, there was an increase at 12 hours but net increase है right for third case. हमारे पास क्या कहते हैं, yes there is an increase बट इवेंचुअली हमारे पास जो स्टेटमेंट है वो क्या कह रहा है एट द एंड ऑफ 24 आवर्स। तो हमारे पास एट द एंड ऑफ 24 आवर्स क्या हो रहा है, हमें कोई डिक्रीज क्या नजर आ रहा है एट एनी गिवन पॉइंट, हम उसके रिस्पेक्ट से यहाँ पर बात कर रहे हैं। सो व्हाट इज वाज देयर एनी डिक्रीज बाय द एंड ऑफ 24 आवर्स, दिस इज व्हाट वी आर चेकिंग। एंड ऑल्सो फॉर दिस क्वेश्चन ना पर्टिकुलरली आप जब थर्ड स्टेटमेंट पर आओगे तो आप रियलाइज करोगे थर्ड स्टेटमेंट आपकी क्या है, इनकरेक्ट है। सो आप सारे वो ऑप्शन जिसमें थर्ड स्टेटमेंट है आप उनको नलिफाई कर दोगे। सो आपके पास बचेगा भी खाली ऑप्शन नंबर बी। Honestly, option one and two aren't your best explanations, aren't your best conclusions, but you for sure know कि थ्री तो सही हो नहीं सकता, तो आप थ्री को तो डेफिनेटली एलिमिनेट करोगे। एंड व्हेन यू एलिमिनेट थ्री आपको ऑटोमेटिकली खाली एक ही ऑप्शन बचेगा और आप उसी ऑप्शन को चूज कर सकते हो। सो इवन दो इफ वन एंड टू डू नॉट मेक डज नॉट मेक सेंस, स्टिल by eliminating थ्री you will get your eventual answer. So हम थ्री को रीड करते हैं। Increasing the concentration of antibiotic always increases the number of resistant bacteria. Now if we look at option over here, antibiotic concentration को देखें हम 600 mg per dm cube में, क्या हमारे पास इंक्रीज इन रेजिस्टेंट पॉपुलेशन हमें नजर आ रहा है केस नंबर थ्री में? केस नंबर थ्री से आई एम टॉकिंग अबाउट दिस वन। Is there any increase in the concentration? Why am I saying concentration? Is there any increase in the population? There's no increase in the population of the resistant bacteria. So we know कि 3 is an incorrect conclusion because जो हमारे पास 600 mg per dm cube है उसमें बिल्कुल भी हमें increase नजर नहीं आ रहा बल्कि उल्टा हमें थोड़ा सा decrease नजर आ रहा है। So we know that 3 is absolutely incorrect and that would eventually give us our answer, option number B. Is this clear? So it was basically a BCQ, best possible choice जो थी आपने उसको चूज किया, which is option number B. ठीक है? What is the correct sequence of events in a primary immune response? T lymphocyte activation, B lymphocyte selection, plasma cell release, antigen presentation by macrophages, cytokines released by T helper cells, B lymphocyte differentiation. This is the correct sequence. Neutrophils do not undergo antigen presentation, T memory cell activation, B memory cell activation हमारे पास कभी भी primary immune response में नहीं होगी. So B is the correct answer. Which statement about the properties of the antigen binding site? The antigen binding site is what? Your variable region in different antibody molecules is correct. They are located on the light chains only. No, हमारे पास अगर आप antibody का structure देखो, so ये हमारे पास क्या कहते हैं? The antigen binding site is basically part of both the heavy chain and the light chain. So A is incorrect. They have a hinge region to give flexibility for different antigens. Incorrect. हमारे पास एक तो एक एंटीबॉडी से एक ही एंटीजन आकर लग सकता है और जो हिंज रीजन है वो जरूर फ्लेक्सिबिलिटी देता है बट एक ही एंटीजन आकर लग सकता है, डिफरेंट एंटीजन नहीं लग सकते. They have binding sites for receptors on phagocytes. That is on the constant region, not on the variable region. They have variable amino acid sequence for different antigens. Yes. हमारे पास when we're saying that the antigen binding site is specific for every antigen, that means कि हमारे पास जो glycoprotein structure है, जो quaternary structure है, that is unique for each different antibody and therefore they're specific in nature. The diagram shows a stage in monoclonal antibody production. X plus cancer cells gives hybridoma. What is represented by X? That would be B lymphocytes. No, 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 not antibodies. That would be B lymphocytes. Antibodies हमारे पास क्या कहते हैं वो सेल नहीं होते ना. हमारे पास हम दो सेल्स को फ्यूज करते हैं तो हम हाँ बेसिकली बी lymphocytes जो हैं उनके जो प्लाज्मा सेल्स होते हैं हम उनको बिल्कुल चूज करते हैं. दैट्स करेक्ट. But antibodies are not cells. They are not cells. They are basically glycoproteins. Huh? That's why D is not our safest answer. It would be B. ठीक है. With that, we conclude this paper.